Hello, my name is Jack Elias. I'm the Dean of Medicine and Biology and Senior Vice President for Health Affairs at Brown University. I have the privilege of working with some great scientists, including Dr. Chun-Gun Lee, Dr. Suchitra Kamali, Dr. Bing Ma, Dr. Bidyo Akisman, Dr. Chang-Min Lee, and Dr. Yang Zhu. We have been studying a group of molecules called the 18-glycosyl hydrolases. One of the prototypic members of this family, or of a subfamily called the chitinase-like proteins, is a molecule, chitinase 3-like 1. This study was focused on trying to figure out the mechanisms that underlie the important role the chitinase 3-like 1 plays in metastatic deposition in the lung. These studies have demonstrated to us that chitinase 3-like 1 is a major regulator of immune checkpoint inhibitors. We have demonstrated that chitinase 3-like 1 regulates PD-1, PD-L1, PD-L2, which form the PD-1-PD-L1 axis. We've also found that chitinase 3-like 1 regulates other immune checkpoint inhibitors, such as LAG-3. These are very novel observations that give a completely new vision for the processes that regulate immune checkpoint inhibition. What we have noticed over the years is that chitinase 3-like 1 plays a critical role in the pathogenesis of cancers when they metastasize to the lung. We have shown that it plays a critical role in lung metastasis from malignant melanoma. We have also seen that it plays a critical role in the pathogenesis of breast cancer metastasis to the lung. What has been discovered over the last 10 years is that the host immune response, the ability of the host to eliminate the tumor, depends on the host being able to mount an immune response against the tumor, and immune checkpoint inhibitors such as PD-1, PD-L1, LAG-3, and so on block the host immune response and augment the growth of the tumors. We have found that chitinase 3-like 1 is the driver of that process, making it a prime target for therapies that would block its ability to augment these inhibitory pathways. When you look at these findings in combination, they clearly demonstrate that chitinase 3-like 1 is a master regulator of these immune checkpoint inhibitors of the host immune response by virtue of their ability to control PD-1, PD-L1, and a variety of other inhibiting moieties. Once we knew that chitinase 3-like 1 was a master regulator of immune checkpoint inhibitors, we asked the very important question: if you inhibit chitinase 3-like 1, can you inhibit the ability of tumors to spread to the lung? We tested this by first generating antibodies against chitinase 3-like 1 and picking out the most potent of these to test. These studies, when we used them in vivo mouse models, showed that when we blocked chitinase 3-like 1, we markedly diminished the ability of malignant melanoma and a variety of other tumors to spread to the lung. We then said, why not block multiple different immune checkpoint pathways at the same time? So we generated antibodies that not just blocked chitinase 3-like 1, but also simultaneously blocked PD-1. We used new methodology, which is the generation of bispecific antibodies. And when we used these antibodies, not only did we get the potent effects that we saw with anti-chitinase 3-like 1, we got a synergistic increase in cell death, and what we're able to demonstrate was a very impressive increase in the generation of cytotoxic T cells that were able to kill the tumor. We believe that we have discovered a master pathway that regulates immune checkpoint inhibitors in the lung and in turn controls the ability of tumor cells to spread to the lung and then grow once they're in the lung. We also believe that we have developed therapeutics in the form of the anti-chitinase 3-like 1 monoclonal antibodies and the bispecific antibodies that are exciting potential therapeutics that can be taken not just into murine models, but hopefully eventually into man to control metastatic disease in the lung and even control primary cancers such as that seen with other circumstances. as we've moved along, everything has gotten more and more and more exciting. We're now at a point where we're absolutely convinced that this is a pathway which we want to be able to control cancer development and cancer spread in people. So we've gone on to develop monospecific and bispecific therapeutics that can hopefully be used in people.